Students, we will proceed further in our chapter Principles and Bioprocesses of Biotechnology. Now we will talk about the cloning vector. So first question comes, what is cloning vector? The genetic material of mostly lower organisms such as bacterial plasmid or viral genome etc. have been exploited as a vehicle to transfer the gene of interest into the specific host to produce a particular phenotype or a, or to produce a particular product. So first we will see what should be the features of a good cloning vector. First one is it must be smaller in size. Means if they will be smaller in size, then it will be easy to introduce that particular vector into the host. The second point is it must be replicating inside the host cell. So if they will be able to replicate by themselves only, then they will autonomously increase their numbers in the host cell. The third point is it must possess restriction site. It means if they will have the restriction site, then it will be easy for the scientists to put the gene of interest in that particular region of that cloning vector. The fourth is introduction of donor DNA fragment must not interfere with the replication property of the vector. It means if suppose we are putting a gene of interest in a vector and that particular gene is interfering with the replication of that particular vector, then the copy number of that vector will not be increased and we will not be able to get the phenotype or product. The fifth point is it must possess some marker genes so that it can be used for later identification and selection of recombinant DNA. It means they should have a particular gene which is called as marker gene. By using that marker gene, we can the identify and select the recombinant DNA into the host cell. The last point is it must possess multiple cloning sites. What is the multiple cloning site? We will discuss this in this lecture. So first is the component of a vector. Means we will discuss what should be the construct or genes to make a vector. So the first component is origin of replication. What is origin of replication? That particular DNA sequence which helps the vector to replicate themselves autonomously into their host cell. The second is multiple cloning sites. We can see here in this vector, there are several restriction sites and the whole restriction site will be called as multiple cloning site. It means we can put our gene of interest here or here or here or here. It means there are several regions where we can cut the DNA or plasmid where we can introduce of gene of interest by our choice. So this is the site of DNA in vector having multiple restriction sites of various restriction endonuclease enzymes as to make a place to insert the gene of interest. The third is antibiotic resistant gene. So we can see here that this vector is having two antibiotic resistant genes. One is ampicillin resistant gene and another is tetracycline resistant gene. So first we will see what is the meaning of this antibiotic resistant gene. These genes help the vector to survive in a particular antibiotic medium, and this sequence allows to survive only those hosts which will have that particular vector. Means we can see here this is the vector and this vector is having two different antibiotic resistant genes, and this vector have been introduced in this population of bacteria, and after introduction of this vector some of the bacteria we can see here that this bacteria and this bacteria have this vector, and this bacteria and this bacteria is empty, means they don't have this vector. And when we have grown the population of this bacteria in the ampicillin or tetracycline, then only those bacterial cell will survive which will have this particular vector. We can see here this one, all the population of bacteria will have will have this vector, and the empty bacteria will be killed because they do not have such genes which will protect them from this antibiotic. So it is very essential to have this segment in the vector. Fourth is marker gene. So these genes help in identification of cells or animals with the vector by showing some phenotypes. For example, green fluorescent protein. So we can see here in first figure A, we can see here there are a total of 6 pups, and out of these 6, 3 are showing green fluorescent protein expression, GFP expression. It means they have the vector, and out of these 6, 3 are 3 are not expressing this GFP protein. It means they do not have such vector. And in this figure, figure B, we can see here that all the cells are expressing green fluorescent protein. So we can identify that particular animal or cell which will have this vector system. So we will discuss some of the cloning vectors. Means we have multiple of options of this vectors. So first is plasmid, and the particular plasmid is PBR 322, and we will see here the structure of this plasmid. We can see here origin of replication, which will help the replication of this particular plasmid. And this is ampicillin resistant gene, this is tetracycline resistant gene, and if this vector is having different restriction sites, it means we can put our gene of interest here in PST1 or EcoR1 site or BAMH1 site or SAL1 or this site. And this is artificial plasmid, and this plasmid has been derived from E. coli bacteria, and the size of this plasmid is 4361 base pairs, and has been used to transfer the construct in bacteria. Means this plasmid has been exploited to produce a product or to create a phenotype in bacteria, and they have the genes as antibiotic resistant gene against tetracycline and ampicillin, and the second gene is multiple cloning sites, different we can see here different restriction sites this plasmid is having. The second is agrobacterium plasmid. So as we know that agrobacterium is a plant pathogen, and it has the ability to transfer the DNA into plant host. That's why the plasmid of this agrobacterium named PI plasmid has been used to transfer the gene of interest into plants. It means if I want to express a particular gene in plant, then I can utilize this PI plasmid. So it is a tumor inducing plasmid, TI plasmid, and it is found in pathogenic species of agrobacterium, Agrobacterium tumefaciens and Agrobacterium rhizobiums. And they have some special gene construct. One is virulence gene. We can see here virulence gene. This gene helps this vector to introduce gene into plant's body. Means it will it will transfer the construct in the plant body. And the second main region is T region. This T region helps this vector to introduce in plant's genome to express their gene of interest. Another example of the vector is RNA virus, or means retrovirus vector, or the genome of retrovirus we can utilize as in the genetic engineering process. So retroviral vectors are derived from the retroviruses. And what are retroviruses? The viruses having genome made up of RNA only. They resembles in the means in case of retroviral vectors, these vectors resemble with the parent virus, except the genome encodes the therapeutic gene or gene of interest instead of viral structural proteins. It means the virulent gene will be replaced by our gene of interest. However, the capsid protein envelop and essentially are essentially identical to the wild type virus, allowing receptor binding, infection, reverse transcription, and transport of the proviral DNA into the nucleus and integration. Means the genome of the particular virus will have all the genes which will help this virus to bind with the receptor of the host in to do the infection, to produce reverse transcriptase enzyme, and to transport the proviral DNA into the nucleus and integration, except the virulent, except the pathogenic gene which is creating a disease. They have genes for capsid protein formation, receptor binding. We have already discussed this. The next is BAC, bacterial artificial chromosome, and we can see the structure of this BAC is a bacterial artificial chromosome is a derivative of bacterial DNA made by genetic engineering. Means by utilizing genetic engineering, the scientists have produced this bacterial artificial chromosome, and these have been used to clone DNA sequences in bacterial cells. Means we can introduce the gene in our gene of interest in E. coli or in some of the bacteria. They can insert genes of size 150 to 350 kilobase pair. We can see here that this is very large size of gene. Means we can put our gene of interest ranges from 150 to 350 kilobase pair also. Often these back vectors have been used in DNA sequencing or genome sequencing of the organism. Since the genome is composed of large stretches of DNA, that's why we used to utilize this vector. Another is YAC, yeast artificial chromosome. We can see the structure of YAC here, and YAC is the derivative of yeast DNA, Saccharomyces cerevisiae, and bacterial plasmid. It means they are also artificially created vector, and it is a combination of some of the yeast DNA as well as bacterial plasmid. And we can see here the insert size is means we can put our gene of interest ranging from 100 to 1000 kilobase pair, and they are again, these vectors have also been used to to uh, in genome sequencing experiments, and they contain genes for antibiotic resistance and visible markers also. We can see here. Thank you.